might not be able to complete this. Let's see. Where is my describe how the sample of chloroplast could be isolated from the leaves. Okay, break open the cells. Okay, homogenize them. Okay, buffer with same water potential. Okay, centrifuge. So being denser or being heavier, the chloroplast would center, yeah, it would be settled at the bottom. Correct. So the main technique is first of all you need to break open the cells, and sometimes we use some chemical which is buffer with the same like pH, the concentration, or maybe the water potential should be same. Then the centrifuge it at a very high speed so that the components can be separated. So chloroplast being heavier, it can be settled at the bottom. So let's move to the next point. The next question is scientists grew two group of plants. Okay, let me just take the question. Okay. The control plant with all inorganic ions needed, whatever potassium, nitrate, or whatever is needed, and the iron deficient plant with all the inorganic ions needed but without iron. Now, the first one has iron, the second one doesn't have iron. After 1 week, the scientists measured the mass of protein and the mass of chlorophyll, and the values are given. The mass of protein is 40 and the mass of chlorophyll is 10. So some protein found inside the chloroplast are synthesized inside the chloroplast. Give one feature of the chloroplast that allow protein to be synthesized inside the chloroplast. What makes protein in chloroplast? What makes protein in chloroplast? What is present in chloroplast? Okay, DNA. What else? Now chloroplast has its own DNA. Which other organelles have their own DNA? The mitochondria. These are the organelles that have their own DNA. So because this have their own DNA, so it can make its like okay its own what protein. Okay, so what is the difference? Is the DNA similar to the nucleus or would be different? How would okay wait? How is this DNA different from the nuclear DNA? The chloroplast DNA and the nuclear DNA. Circular. Okay. Is linear, and moreover they have the histone protein and other things as well. Okay, if you want to mention the other one organelle, it can be ribosomes, and the ribosomes are much smaller in these organelles as compared to the cytoplasm. Whatever ribosome are present in cytoplasm, they're bigger in size. Okay, what is the ratio of protein to chlorophyll in oh, okay, the ratio is given, already given. Use this information to fill in the in table 4 and calculate the ratio of protein to chlorophyll in iron deficient plants. They have already given us the ratio, like it's a 40 for mass of protein and percentage of control, and it is 10. So how can you do this? We need to put in numbers. How would you do this? Suggest anyway. Like, okay, wait, let me. So they have given you, wait. They have given you a ratio of mass of protein divided by percentage of control. Now what you need to do, you need to calculate the ratio of protein to chlorophyll. Here you have to mention protein, and here you have to mention chlorophyll. So the ratio is 4 ratio 1 is 40 ratio 10. For example, we are saying, let's say 40 ratio 10. So how would you figure it out? If it's 9 ratio 1, wait, I'm telling you. Okay, let's do it. It would become 4 ratio 1. Is it so? 4 into 9 is 36. That would be your point. 1 into 1 is 1. 36 ratio 1. Correct. Okay, so this is all for this paper. We'll discuss the other questions in the next video, or in the, sorry, next lecture. So see you on Friday, and on Friday we would have the last lesson, I guess. So if you have any confusion, you can ask me on Friday. We'll try to solve it. And I would really appreciate if you send me the questions at least an hour before, so I can go through those questions. Okay? Thank you so much. Have a great day. Goodbye.